I want to talk about a few genes that affect the human developmental pathway. This is taken from the book. The picture here is on page 91 of the text. So SRY is a gene on the Y chromosome, and its expression is a transcription factor affecting a genetic cascade and ultimately turning on and turning off a large number of genes involved in differentiation, sex differentiation in humans, as the text discusses. So one effect of this is to promote the formation or stabilization of the Wolffian ducts, and the Mullerian duct degenerates in males in anyone expressing SRY. And it's an odd figure, actually. I mean, it goes here with testis and cholesterol, and they're not really parallel things, are they? But you get the idea of the flow here. And so the first gene I want to mention is CYP1, or I'm sorry, CYP21. And that is connected with a particular condition, or mutation in that gene is connected with a particular condition referred to as congenital adrenal hyperplasia. But the effect is, so that the normal function of the protein product of CYP21 is to reduce testosterone and DHT levels. And so if you have a mutation in that gene, then what's going to happen is it no longer reduces those levels the way they're supposed to be. And so, as a result, someone with a mutation in CYP21 has higher than normal levels of testosterone. So this is affecting the pathway here before the formation of testosterone. And so as a result, males with this condition, males who have the CYP21 mutation, have hypermasculinization, I guess, early puberty, increased musculature, and things like this. And similarly, females who have the condition also have some, it's counterbalanced by other factors, but they do have some hypermasculinization as well. Although they are anatomically female, they still do have some male secondary characteristics, deepening voice, facial hair, things like that. So that's the effect of one mutation on this pathway. And the second gene I want to mention is called SRD5A2. Great names, SRD5A2. We got some great names here in human gene land, unlike Drosophila, where they've got some interesting names at least. So SRD5A2 is a gene whose protein product is involved in this process, but it acts at a different point. SRD5A2 is involved in the conversion of testosterone to DHT, dihydrotestosterone. And so if you have a mutation in that gene, you're going to have some problem here with converting testosterone to dihydrotestosterone. DHT, it's why they call it DHT, I guess. And so what's going to happen then is your testosterone levels are unaffected initially. I mean, ultimately they'll go up because you're not transmitting or changing the testosterone to DHT. So the testosterone levels are normal. Androgen-sensitive cells, those cells in the body which respond to testosterone, are going to function normally, and you're going to get normal internal male structures in individuals with these mutations. However, DHT, dihydrotestosterone, is involved in interacting with other androgen-sensitive cells, and they're going to be responsible for external male structures. And those are going to be defective because you haven't produced as much DHT as you're supposed to, and so that's going to affect the formation of external male structures. And so, in fact, individuals with this mutation appear to be female because they don't have the external male structures have not developed normally. So babies are born, they appear to be female. However, at puberty, you get testosterone. That's when testosterone really kicks in, and you get, again, secondary sexual characteristics, deepening voice, facial hair growth, beginning despite the external female genital structures. And note that in the figure in the text, they swapped the little balloons indicating which is which. The genes were shown acting at the right points in the pathway, but the text is correct. The image is not, as far as CYP21 leads to congenital adrenal hyperplasia, and SRD5A2 mutations lead to 5-alpha reductase deficiencies, unlike what the figure says in the book. The third gene that I want to mention is simply called AR, for androgen receptor. And so what this is These androgen-sensitive cells are, as I alluded to before, responsive to androgens, testosterone, dihydrotestosterone. If you have a mutation in AR, then it's going to affect these cells. And so they're not going to be, if they're AR minus, they're not going to be responsive to testosterone DHT. Those hormones are made. They're made at normal levels. They circulate through the bloodstream. Everything's fine as far as that's concerned, but the cells basically are sitting with their fingers in their ears going la 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 la, not paying attention. And so as a result, you have some very serious effects here. Again, the default pathway in humans is female, and so individuals with AR mutations well, I say individuals. A woman with an AR mutation probably wouldn't have a whole lot of effect. I'm not a clinician, I don't know the details. But if you are genetically male, if you have an X and a Y chromosome, you should be male. Instead, you're going to appear again female, despite the fact that you're producing normal amounts of testosterone and DHT. So you appear to be, you would appear to be female, but internally your reproductive structures don't develop either as male or female. Because what happens is the insensitivity prevents the development of the male sexual structures internally and externally, for that matter. So you don't have the plumbing, if you will, for male sexual reproduction. But SRY is nonetheless functional, and it results in the degeneration of the Müllerian duct, and so the female structures aren't present either. So the reason I'm going over this is just to illustrate, in a somewhat interesting way perhaps for some people, some of the complexities. This is just one of many, many, many hierarchical pathways involving multiple genes with multiple effects in different dimensions and different directions. And so I hope this was somewhat interesting. And that's it.